with this presentation, I just want to briefly go over some of the research projects that have been funded by the LMI. And these research projects are focused on Gorham-Stout disease, GLA, and KLA. Before we discuss the research, I just want to briefly introduce or go over the history of the LMI. The LMI is a nonprofit organization whose mission is to improve the care of patients with GSD, GLA, or KLA. And to carry out this mission, we foster collaboration among investigators working on these diseases. We help investigators develop research tools. We organize scientific meetings on GLA, GSD, and KLA. And we fund research on these diseases. And our funding model is unique and different from other organizations. It's a very interactive model where the LMI interacts with investigators and helps them develop their research questions. And this ensures that the goals of the research project are in line with our mission. And it makes sure that the research that's being done is being done with the hope of identifying a therapy or a better understanding of GLA, GSD, or KLA. And we've funded over 19 researchers, and since 2011, the LMI has invested over $3.5 million in research on these diseases. And our research program is broken up into 3 distinct areas. So we fund molecular studies, cell-based research, and also preclinical and clinical research. And the first area that I'm going to go over is our molecular research, which is focused on DNA, RNA, and proteins. So we're very interested in identifying the genetic mutations that cause GLA, GSD, and KLA. And we have funded 3 research teams from around the world, actually, to identify mutations in patients. So the first team is made up of Juan Carlos Lopez Gutierrez and Victor Martinez Glez, and they've been working on identifying mutations in GLA patients. Mika is working with TUI, and also Shoshana Greenberger is in Israel, and she is the person that has identified an NRAS mutation in a patient. So these teams are working with samples that they have at their institutions, and we're also working together and interacting with one another and sharing our information that we gather from the sequencing studies. So we think that this team will help us identify the genetic mutations that cause these diseases. The LMI also funds cell-based research, and I'm just going to discuss one of the research projects that we have funded. So we're interested in developing a better understanding of how lymphatic endothelial cells could induce the destruction of bone. And the investigator that we funded is Lanping Xing at University of Rochester. And she was interested in understanding how lymphatic endothelial cells affect the activity of osteoblasts and osteoclasts. So if you remember from the presentation earlier today, osteoblasts are cells that synthesize bone, and osteoclasts are cells that degrade bone. And what Lanping has found is that lymphatic endothelial cells stimulate the development and activity of osteoclasts. They don't appear to affect the activity or development of osteoblasts. So her working model is that when lymphatic endothelial cells arise in bone, they stimulate the development and activity of osteoclasts. And we think that this helps promote bone loss in patients with bone involvement. We've also funded preclinical and clinical research projects, and much of the research in this area has been focused on developing animal models of GLA, GSD, and KLA. And most of our models are actually mouse models. And mice are frequently used to generate models of human disease. Mouse models are useful for research because they allow investigators to closely control their experiments. And mice are really an unlimited supply, so if we need more mice, we just simply breed up more, and we can get more mice for our studies. So mouse models have improved our understanding of the progression and cause of other human diseases, and we believe that we could use mouse models to develop a better understanding of GLA, GSD, and KLA. And one of our investigators, Donald McDonald at UCSF, has developed mice that develop irregular lymphatics in the lungs. So we were using this as sort of an animal model of pulmonary involvement in GLA or any of the lymphatic anomalies. So he has mice that develop irregular lymphatics in lung, and he was using these mice to test the effect of various therapies. And his goal was to try to identify a drug that could reverse or normalize these irregular lymphatics that are present in the lung of his mice. And he tested several different drugs and found that rapamycin could partially normalize these irregular lymphatics in lung. We have research going on in my laboratory here at UT Southwestern in Dallas, and we set out to create an animal model of Gorham-Stout disease. And we created transgenic mice that express this factor in their bones, and this factor stimulates the formation of lymphatic vessels. So we thought we could create mice and trick their bones into developing lymphatics. So what we're looking at here is sections of bone from a normal mouse and a Gorham-Stout mouse, and we've stained these sections with an antibody that detects lymphatic vessels. And you can see in the normal mouse there's no brown, there are no lymphatic vessels in bones from a normal mouse. But with our Gorham-Stout model, we can see that the bones from these mice are actually filled with lymphatic vessels. So we're using these mice to develop a better understanding of how lymphatics actually get into bone. When we look at the structure of the bones from our mice, we can see first on the left you can see what normal bones look like, what normal femurs and ribs look like. And you can see our animal model, the mice have very porous bones. So we're also using these mice to develop a better understanding of how lymphatic vessels promote bone loss. There's research being done to identify biomarkers for lymphatic anomalies, and I just want to briefly go over what a biomarker is. So a biomarker is a measurable indicator of the severity or presence of a disease state, and there are several different types of biomarkers. Some biomarkers are called diagnostic biomarkers, and these biomarkers assist in the diagnosis of a particular disease. So this is something that we could assess, and then that would help us determine which disease you have. There are also prognostic biomarkers which carry information on the course of the disease. Is it going to be severe or mild? And there are predictive biomarkers which help predict how a patient could respond to a particular therapy. And biomarkers could also be used to monitor the response of a therapy. And what we're really searching for is a circulating biomarker, something that we could measure non-invasively, something where we could collect blood from a patient and measure it, and then that would give us information that would help with the diagnosis and treatment of the disease. And we have funded a project that's being led by Denise Adams and Tim LaCroix. And what they have found is there's a particular factor in the blood of KLA patients that's very high. And it's higher in KLA patients than GLA patients, GSD patients, or other lymphatic anomaly patients. And this factor is called angiopoietin 2. And we think that this factor is going to assist with the diagnosis of KLA, where based on how the clinical presentation, but then also the measurement of Ang2, you could achieve the correct diagnosis of KLA for these patients. They're also monitoring Ang2 to have a measurable indicator to response to therapy. And what they have found that is after treatment with sirolimus, that the Ang2 levels decrease in patients as their disease improves. So we think that we could not only use Ang2 to help diagnose KLA, but also to monitor response to therapy. One of the first projects that we funded was led by Cameron Trenner, and he created the Lymphatic Anomalies Registry. And this is a registry that's we hope will help us develop a better understanding of the natural history of lymphatic anomalies and also to evaluate the effect of various therapies on patient outcomes. And the website for this registry is located there on the bottom of this page, so lymphaticregistry.org. So we encourage patients to actually participate in both the LGDA registry as well as the lymphatic anomalies registry. Another one of our goals was to collect patient samples and have a little biorepository that we could share with other investigators that are doing research on lymphatic anomalies. And we worked together with Eric Eklund, and he's created a small biorepository of patient samples. And these samples are formalin-fixed, paraffin-embedded samples, so not the fresh samples or frozen samples that Tui talked about. And he's been very collaborative and willing to share the samples that he's collected with other investigators, and this has really helped us move a few research projects forward. So in addition to funding research, we've also organized international meetings on GLA and GSD. Our first meeting was in Bethesda in 2013, and our second meeting was just 2 years ago in Atlanta. And the objectives of these meetings were really to bring together leaders in the field and have them interact with one another and form collaborations and new avenues of research. And the reports for these meetings are published and are available. So the projects that we have funded have resulted in several publications. So this shows that the projects that we are funding, the results from them are going out to the scientific community to where they can have an effect. And we've published so far 13 papers, and we anticipate that 3 or 4 more will be published this year. So with respect to the future, we hope to identify the genetic mutations that cause GSD, GLA, and KLA, and I think that that can be done within the next few years. This knowledge will help guide the search for therapies, establish new animal models of these diseases, and it will really open up the field to new investigators, and particularly patients that don't have access to tissue samples. So to conclude, our understanding of the features of lymphatic anomalies have really increased since Gorham and Stout published their paper in 1955. However, many questions still exist, but importantly, numerous research projects are underway to address these basic science and clinical research questions, and we believe that these research efforts will lead to a deeper understanding of the cause of these lymphatic anomalies and will help identify effective therapies. Thank you for your attention, and you can learn more about the LMI and the research projects that we have funded by visiting our website. Thank you.